All right, we're going to be talking about mitosis today, and one of the main events that happen with mitosis is the skin cells and stomach cells. This process only makes body cells. It does not make eggs or sperm. It starts with interphase. Interphase is the division of when the cells duplicate the chromosomes. Most of the time, the cells do stay in interphase division most of their life. Within mitosis prophase, that is when the chromosomes will line up. They do contain homologous pairs. They do contain the same size and the same shape as the parent chromosome. It means it could be the same type of gene no matter what. If it's the parent chromosome. Prophase within mitosis, that is where the chromosomes start coming apart from the center of the cell, and then they line up along the metaphase plate to make sure the chromosomes are all lined up correctly. These do line up within a straight line. It does not matter which order. Some can be on the top, some can be on the bottom, but most of the time they all will line up in some sort of single file line down the center. Of the metaphase plate. In this phase of the metaphase cycle within mitosis, the nuclear envelope has been disassembled, so then the meiotic spindles can go ahead and start pulling the chromosomes apart. With the meiotic spindles. They're like little threads that pull these apart. So they start out in the middle in a single file line, which is like this. Then the meiotic spindles, which is this pink thing right here, is going to be my meiotic spindle. That pulls out some threads and it will pull this chromosome this way, and it can pull this one this way. That is what happens during meiosis. Mitosis, sorry. In the metaphase section. Within mitosis, the chromosomes line up in pairs within metaphase 2, but that has to deal with meiosis. Now we're moving on to mitosis within anaphase. This is where the chromosomes will move away from each other from the metaphase plate. The chromatids are pulled away by the work of the spindles. So these chromosomes that were lined up all together here. Let's see, here's another pair. So these are both lined up on the metaphasal plate. This is my spindle, this pink thing right now. So that will be the one that's in charge of pulling these chromosomes away from the center of the cell. Those spindles are what push and pull these chromosomes apart, these chromatids, apart from the center of the cell. Next, you will have telophase. That is where the chromosomes will be lined up. So you will have a bunch over here, all scrunched together, and you will have some over here. But each of these will be on separate sides of the cell. On separate sides of the cell, these during telophase. Then they begin to form a nuclear envelope around these cells, these chromatids, to then form. So let's say we'll use this as an example. This is the cell, and the chromatids are inside this cell. Like this. And here's our cell, the outside here, and then the chromatids will be inside. This is the outside of the cell. This is the chromatids inside the cell. These are going to form nuclei on each side. So each one of these pairs of chromosomes will have their own nuclei around them, making their own cell, their own daughter cell. But one DNA is from one parent and one DNA strand is from another parent. And when they cross over, they're exchanging their DNA information. That is how they exchange that information, is through the crossing over action. That's how they exchange DNA, and they're able to form new daughter cells. Then they form, after telophase, you have cytokinesis. That is when the cytoplasm is dividing, and then that will divide the chromosomes into their own type of cell. These are identical within this process. They are diploid. They do contain two sets of chromosomes, one from each parent, and they also are mainly used for repair or just making new skin cells or body cells that we lose over time. But this is the process of mitosis